Today's topic is membrane transport. How ions, macromolecules, gases are transported across the membrane. All cells, either prokaryotes or eukaryotes, are separated from their surrounding environment by plasma membrane. In eukaryotic cell, we are having compartmentalized cells, like compartmentalization is there. And we all know that these biological membranes are semi-permeable in nature. That is, they allow specific molecules to enter and waste products to leave the cell. The movement of these specific molecules, like we can talk about the sodium, potassium, calcium, or chlorine, amino acids, sugars, and even also water. So there are specific transportation system in this plasma membrane. Plasma membrane has ability to maintain its constant internal environment. That is, it is able to maintain in a homeostasis stage. Even within in the cell, we are having different ion concentration. If we talk about cytosol to the organelle, we are having different ion concentration. Like in case of lysosomes are having higher concentration of proton, H positive ions, and it varies from cytosol up to 100 to 1,000 times. It means that the lysosome are having higher concentration of protons as compared to its cytosol. So every plasma membrane of every organelle has its specific function, and this specific function is allowing cell to perform different functions, and this is all because of the permeases or the protein transport which are present on this plasma membrane. We know that our plasma membrane is semi-permeable, as you can see in the diagram. Like gases, if we talk about the carbon dioxide, oxygen, they are directly passing through our plasma membrane. Or even if we talk about the ethanol, that is small uncharged polar molecule, this ethanol is also having ability to pass through the plasma membrane. Or if we talk about the urea or water, they are permeable to some extent, not that much. Or if we talk about the larger uncharged polar molecules, glucose or fructose, they are not directly allowed to pass through the membrane. So we need a permeases or transport proteins which will allow them to cross the plasma membrane. We are having ions, potassium, magnesium, calcium, chlorine ions. They also need transport system to get into the cell. Or we are having charged polar molecules. ATP is there, glucose 6-phosphoproteins, amino acids. So all these molecules need permeases to cross the plasma membrane. As I said, that the concentration of ions vary within in the cell as compared to its external environment. As if we talk about the cations and the anions, they vary. As the concentration of sodium in an intracellular within in the cell, it varies from 5 to 15 millimolar. So that is, sodium cations are within in the cell in the concentration of 5 to 15 millimolar, and extracellular concentration of the sodium ions are 145 millimolar. Magnesium ions are in the range of 30 millimolar within the cell, or outside the cell it's 1 to 2 millimolar. Calcium ions, intracellular concentration of calcium ion is in the range of 1 to 2 millimolar, and extracellular concentration of calcium ion is 2.5 to 5 millimolar. Or we talk about the chlorine anions, the intracellular concentration is 4 millimolar, and the cellular concentration is 110 millimolar. So it means the cell is having ability to maintain its internal constant environment. It's not that whatever the molecules are present outside the cell should be present in a same concentration within the cell. That is, we call that plasma membrane is able to maintain its internal environment, and this constant internal environment is called as homeostasis. Now, how these proteins or how these molecules are transported across the plasma membrane? They can simply diffuse some components. They can simply diffuse our lipid bilayer membrane. Such type of protein is called as simple diffusion. Or these components or the compounds need tunnel mediated or carrier mediated. All these processes come under the passive transport system. Passive transport is the transportation of the molecules from inside to outside or outside to inside without the utilization of ATP as an energy source. Without the utilization of ATP as an energy source. And another type is our active transport, where ATP is utilized and which takes molecules from its lower concentration to higher concentration. It means against the gradient. Now we will discuss passive transport in detail. So passive transportation can occur by three different processes. It can occur by osmosis, simple diffusion, facilitated diffusion. It can occur by osmosis, simple diffusion, and facilitated diffusion. Now osmosis. This semi-permeable membrane is having ability to move water from lower salt concentration to higher salt concentration. Or we can say water moves from higher concentration to lower concentration. That is the solution. That is, in other words, we can say the solution of higher water potential to a region of lower water potential. So there is the method. It means water is sometimes entering into the cell or water is moving outside the cell. If the water molecules are entering into the cell, it is endosmosis. Or if this water molecule is moving outside the cell, it's exoosmosis. So we can, as you can see the diagram, we are having three different types of solutions: hypotonic solution, isotonic solution, and hypertonic solution. Hypotonic, isotonic, and hypertonic solution. First, we will discuss about the isotonic solution. As you can see, the water with the yellow arrow is moving in and the gray arrow, water is moving out of the cell. It means the concentration of ions within the cell and the concentration of ions in the solution is same. That type of solution where the cell is neither able to lose its water or gain its water. So they are in a such type of solutions are called as an isotonic solution, जहाँ पे पानी अंदर भी आ रहा है, बाहर भी जा रहा है, and the concentration of the solution and the cell is same. For this isotonic solution, we always make 0.9% NaCl. 0.9% NaCl, sodium chloride solution. So we can place our erythrocyte cells there. In 0.9% solution, neither our cells will get burst up by taking excess of water, neither they will shrink. When the solute or the water concentration outside the cell is, when the water concentration outside the cell is less, that is, they are having higher concentration of solute. So at that time, water is going to move from cell to the external environment. Such type of solution is called as hypertonic solution. पानी सेल से बाहर के environment में आ रहा है. क्यों? जो बाहर का हमारा environment है, that is having higher concentration of solute. Or if we talk about the hypotonic solution, it means the ion concentration or the solute concentration within the cell is less. as compared is less as नहीं is more as compared to the outside cell. Cell के अंदर solute या ions की concentration ज़्यादा है as compared to the environment. So outside it means water की concentration ज़्यादा है. So water is coming into the cell. That time there is a rupture. Cell will get ruptured if it's having excess of water in it. So osmosis can be studied via the three processes like isotonic solution, hypertonic, and hypertonic solution. Second, simple diffusion or diffusion. It is the movement of solutes from a region of higher concentration to a lower concentration. As you can see here, that the solutes on extracellular space is having higher concentration. Then with the help of simple diffusion, they are passing through this plasma membrane and coming into the intracellular space. Higher concentration of solutes moving to the lower concentration of solute is called as diffusion. And the difference in concentration between the two regions is called as concentration gradient. It means when there is a difference in the concentration of solutes, एक जगह ज़्यादा solute है, दूसरी जगह कम है. It means there is a gradient formation, कम और ज़्यादा. So this such type of difference in concentration is called as concentration gradient. And this diffusion is this diffusion is occurring till that time there is no difference in the gradient, or this gradient get vanished up, or we can say the solutes are coming in that when there is a distribution of solute equally, like outside the cell or inside the cell, solutes are equally distributed. At that time, then this diffusion stops. As you can see, the diffusion stops. It means equal ions or equal concentration of solute is now present either on extracellular space or intracellular space. So simple diffusion can occur for oxygen, carbon dioxide, or ethanol molecule. Now the third part of our acid transport is facilitated diffusion. This type of diffusion, or the movement of molecules across the plasma membrane, they are carried by the special type of transport proteins. These, as you can see in the diagram, these proteins are embedded or within in the plasma membrane, through which this facilitated diffusion occurs. Again, I will repeat, in facilitated diffusion, there is the level like ions or molecules are transported via the proteins or permeases. But again, it's a passive type of diffusion. It means there is no utilization of energy. Like this process can occur when large molecules such as glucose, these glucose molecules bind to specific carrier molecules, and they are having the receptors on these carrier molecules. Then these carrier molecules, the glucose is taken from extracellular space into the intracellular space. This facilitated diffusion is having specific characters. As we can say that this facilitated diffusion is faster. The rate of diffusion is faster as compared to the simple diffusion. This transportation of molecules from extracellular space into the intracellular space is much faster or greater. And this is specific. Why? Because we are having channel proteins there. They only carry specific species of or molecules, because as according to their size, whatever fits their size, receptor is there. This receptor is having like a receptor site. So this receptor site will accept only few molecules. They will not accept all the ions or all the molecules which are present in the extracellular space. Depends on the shape of the receptor. So that the molecule which is having same shape as to the receptor site will get only transported. It means this facilitated diffusion is specific in its activity. So they carry only specific molecules according to their as per the shape of the surface receptor of protein. For this, we are having best example that glucose is transported into the erythrocyte cells. That is, D-glucose permeases of erythrocyte. Or we can say the movement of small molecules across the membrane. The mammalian erythrocyte are having these type of channel proteins. These channel proteins are involved in the facilitated diffusion, which takes glucose from extracellular space into the intracellular space. And these proteins, which are involved in this glucose transportation, we call them as glucose transporters. We can call them as glucose permeases, or we can call them as D-hexospermease. And the glucose is transported into the erythrocyte cell, then within no time, this glucose is getting phosphorylated by the enzyme hexokinase. You have studied this in the glycolysis cycle. So it means glucose is getting converted to glucose 6-phosphate by the phosphorylation, and this phosphorylation activity is carried by the enzyme hexokinase enzyme, and it utilizes 81 ATP there. That means from ATP, phosphate group is transferred to the glucose, which makes our glucose as a glucose 6-phosphate. When glucose is getting converted to glucose 6-phosphate, it means there is always lower concentration of glucose within the cell, because its simple glucose is getting converted to some other form. So it means inside the RBC cells, we are having less glucose, as this glucose is getting continuously converted to glucose 6-phosphate. So we have a concentration gradient. It means every time, every time the glucose is coming into the erythrocyte cells via the simple diffusion, because this glucose is no longer in the glucose form, they are getting converted to glucose 6-phosphate. And all plasma membranes except mitochondrial membrane, no plasma membrane is having the ability to pass these phosphorylated compounds. When the cell has to retain its simple compounds, so these compounds these compounds are getting converted, or these are getting converted by the process of phosphorylation reaction. Means ATP हमेशा उनके साथ attached हो जाता है, फिर ये जो molecules होते हैं ये फिर वापस नहीं जाते हैं अगर in case for example बाहर वाले extracellular environment में ग्लूकोज की कमी रहेगी, लेकिन ये ग्लूकोज में ability नहीं रहती है to pass through these plasma membrane, because this glucose is now in the form of glucose 6-phosphate. Except I talk about the mitochondria, means mitochondrial membrane is having the ability to move such type of molecules. This D-glucose permease or glucose permease is an integral membrane protein. It's estimated at the molecular weight. The molecular weight of these integral membrane protein is 45,000 daltons, 45,000 daltons, or 45K daltons. And if we talk about them, as I have said earlier, that every plasma membrane or every cell is having their specific membrane proteins. So these D-glucose permeases accounts for 2% of erythrocyte membrane. जो हमारी erythrocyte membrane है, उसमें से जो हमारे 100% proteins है उसमें present, उसमें से 2% कौन बनाता है? इसमें से 2% बनाता है हमारे D-hexopermeases. Now the second type of transportation is our active transportation. It is the transportation of molecules across the plasma membrane against its concentration gradient. So if we are doing something against the concentration gradient, that time ATP is utilized, energy sources utilized to drag these molecules against the concentration gradient. We are having many inorganic ions, amino acids, carbohydrates, which are actively transported across the membrane. And the best example is how we will be able to study this sodium, potassium, how we will be able to study this active transport. Is best example is the sodium potassium pump, these pumps are called as ATPases. Sodium, potassium, ATPases. As you can see the diagram. Sodium potassium pump. It means these ions, or we can call them a cation exchanger pump, or we call them as an ion pump also. With the help of the energy, ATP energy, they are exporting 3 sodium molecules outside, means from sodium is thrown from inside to the outside environment, and they are importing 2 potassium ions inside the cell. 3 sodium ions को बाहर भेज रहे और 2 sodium ions को अंदर ला रहे. This sodium potassium ATP
potassium ions को अंदर ला रहे, सोडियम को बाहर भेज रहे.
Which is best studied in the eels. These eels are having electrical organs, and these eels are very rich in this sodium potassium pump. इसमें बहुत ज़्यादा sodium potassium pump रहता है. That is why it was easy for scientists to study these. So they have observed that the sodium potassium pump is having 2 subunits. As you can see the chocolate color, this is the 2 subunits. One is the larger subunit and one is the smaller subunit. The smaller subunit is glycoprotein. Smaller subunit is glycoprotein, and the molecular weight of the smaller subunit is 50,000 daltons. And it's said that its function is still unknown. If we talk about the larger subunit, its molecular weight is 1,20,000 dalton. And this is the subunit which is responsible for this activity like sodium potassium exchange, or this is the subunit which actually perform this cation transport. So when active transport is going to happen, so on the inner side of this pump, 3 sodium and ATP are getting bind on this pump. Then ATP hydrolysis takes place, which phosphorylates this pump, protein pump. Due to this phosphorylation, there is change in the shape. जब क्या हो रहा है, 3 सोडियम अटैच हो रहे हैं और ATP हो रहा है अटैच. ATP हो रहा है स्मॉलर सब यूनिट पे अटैच हो रहा है. तो फिर ये जो ATP है हाइड्रोलिसिस दे रहा है, ये अपना इनऑर्गेनिक फॉस्फेट किसको दे रहा है, स्मॉलर सब यूनिट को. जो ये एनर्जी आ गई, इस एनर्जी की वजह से क्या हो रहा है, जो ये हमारा प्रोटीन पंप है, इट चेंज इट्स शेप. देन ड्यू टू चेंज इन इट्स शेप, दे आर एबल टू रिलीज सोडियम आउटसाइड द सेल. पहले अटैच हो गए, फिर हाइड्रोलिसिस हो गया किसका, ATP का. Then due to this hydrolysis, this pump or the protein or the permease change its shape, due to which the sodium ions are thrown out of the cell. And this change in the shape enables potassium ions to bind onto the pump. जो ये change हो गई shape, वहां पे एक ऊपर receptor बनता है जहां पे sodium potassium ions attach हो जाते हैं. Potassium ions attach हो गए, then there is a release or the removal of the inorganic phosphate or release of the inorganic phosphate from the pump. जब inorganic phosphate release हो गया, तो इस pump को क्या है, अपनी original shape में आना है. So while coming into its original shape, the 2 potassium ions which was attached on the outer surface are thrown into or releasing into the cell. That is, they are exporting this potassium ions from outside environment into the intracellular space. This is one of the best example of active transport. This is how the transportation of molecules takes place by the active transport mechanism. We are having 3 different forms of active transport: Uniport, Symport, or Antiport. Uniport means एक जगह से molecule आ रहा है, continuously अंदर आ रहा है. Means the molecules are continuously coming from outside to inside. That this is such type of transportation is called as Uniport. Means वहाँ पे single direction है. In Symport, what is happening? 2 molecules counter to each other are coming in as compound A and compound B. They are coming together. This is Symport. And the antiport, 1 molecule is coming in and 1 molecule is going out. Symport, now we can say, Symport uses a downhill movement of one solute molecule from its higher concentration to move another molecule uphill from its lower concentration to higher concentration. That is, it's against its electrochemical gradient. This is a symport, and in case of antiport, two species of ions are pumped in opposite direction across the membrane. जो two species of ions are pumped across the membrane. That is, it allows the flow from higher to lower concentration. And the best example we have studied just now is our sodium calcium or sodium potassium exchanger. Now we have studied the passive transport. All these processes are happening because of the presence of the proteins which are present on the plasma membrane.